Polymerase chain reaction, PCR. In this video we will focus on definition of polymerase chain reaction, PCR, PCR primers, oligonucleotide, PCR reaction setup and PCR reaction. Definition of polymerase chain reaction. A cell-free method that utilizes a heat-stable DNA polymerase to synthesize copies, amplification, of a small predetermined DNA segment of interest within a complex starting DNA material. PCR was developed by American biochemist Kary Mullis in 1983. He was awarded the Nobel Prize in Biochemistry in 1993 for his work in developing the method. PCR primers, oligonucleotide. A strand of short nucleic acid sequence that serves as a starting point for DNA synthesis complementary to both ends of the targeted DNA fragment. Length, 18 to 25 bp. Base composition, GC content 40 to 60%. 3-terminal sequence, 3 end of 1 primer, should not be complementary to any region on the second primer. Self-complementary, inverted repeat and self-complementary sequence should be avoided. DNA extraction and purification. DNA isolation protocols comprise of the basic steps of disruption of the cell wall, cell membrane and nuclear membrane, to release the DNA into solution, followed by precipitation of DNA while ensuring removal of the contaminating biomolecules, such as the proteins, polysaccharides, lipids, phenols and other secondary metabolites. PCR reaction setup. A sample consists the following ingredients. H2O. PCR buffer, stabilizes the DNA polymerase, DNA, and nucleotides. KCl and tris HCl. Primer, forward and reverse primers. A strand of short nucleic acid sequence that serves as a starting point for DNA synthesis, complementary to both ends of the targeted DNA fragment. DNTPS, deoxynucleoside triphosphate, DNTPS. Single unit of the bases A, T, G and C, which are essentially building blocks for new DNA. MgCl2, essential cofactor of DNA polymerase. Too little, enzyme won't work. Stabilizes the DNA double helix. Too much, DNA extra-stable, nonspecific priming. DNA polymerase. DNA template. Tubes with the samples be placed in the thermal cycler to start the reaction. The polymerase chain reaction. First, the mixture is heated. To separate the DNA strands, 2 different specific oligonucleotide primers are added that are complementary to short sequence stretches on either side of the desired fragment. After lowering the temperature, the primers hybridized to the DNA, where they bind specifically to the ends of the desired target sequence. A heat-stable DNA polymerase and nucleotide triphosphates are added. The polymerase extends the primers and synthesize this new complementary DNA strands. At the end of this first cycle, 2 double-stranded DNA molecules are produced that contain the target sequence. This cycle of events is repeated. The mixture is again heated to melt double strand of DNA. The primers are hybridized, and the DNA polymerase synthesizes new complementary strands. At the end of the second cycle, for a double strand of DNA molecules are produced that contain the target sequence. In the third cycle the mixture is heated. The primers are hybridized, and DNA polymerase synthesizes new complementary strands. At the end of the third cycle, 8 double-stranded DNA molecules are produced that contain the target sequence. 2 of these molecules are precisely the length of the target sequence. In future cycles, this population increases exponentially. Cycle 4 heating. Hybridization. DNA synthesis. At the end of the 5th cycle, There are 22 double-stranded DNA fragments, of the correct length, and 10 longer ones. Cycle 6. Cycle 10. Cycle 15. Cycle 20. After 30 cycles, there are over 1 billion fragments, of the correct length but only 60 longer ones. We have discussed in this video, definition of polymerase chain reaction, PCR primers, PCR reaction setup, and PCR reaction.